one antibody defect that does have important ramifications for patients is called hyper-IgE syndrome. So again, let's take a step back and look at this as clinicians. So we have a patient who comes into the clinic presenting with a set of symptoms. So we see in their blood they have very high IgE. They also have recurrent skin and lung bacterial infections, as well as chronic fungal infections and chronic dermatitis. They also have skin boils. Okay, so when we do blood analysis of cytokines, we see that there's an increase in pro-inflammatory cytokines expressed in leukocytes. And if we think about why this might be, we can do some genetic analysis and see that there's actually a Th1 defect. And Th1 is important for cytokine signaling, which we'll talk about a little later. So there's one other type of hyper-IgE syndrome that has a separate genetic defect. So again, some of these symptoms are similar, but then we also see an increase in allergies and asthma. So here we see that the immune cells in these patients actually have a diminished ability to move through tissues. And this is due to a Doc8 defect. And Doc8 is really important for the actin cytoskeleton, and as well as MyG88 signaling. So, as we said, that IgE syndrome can be caused by a Th1 defect or a Doc8 defect. And this leads to reduced Th17 response and reduced antimicrobial resistance of epithelial cells. We also have a loss of barrier function, which is how we see the chronic dermatitis and boils. This is partially due to a reduced response to chemokines. So as you guys talked about in earlier lectures, chemokines can lead to epithelial cells producing more antimicrobial peptides. So if they cannot respond to those cytokines, this will not happen. There's also a deficiency in actin cytoskeleton regulation caused by Doc8, and this prevents cells from being able to extravasate through the epithelial layer. So interestingly, the exact cause of the increase in IgE is unknown. So we can explain a lot of these symptoms, but the high blood IgE we still don't understand. So as you can see, there are several antibody deficiencies, and we'll go through a couple more just briefly so you can see them. So as we talked about, defects in CD40 ligand can lead to X-linked hyper-IgE syndrome, as well then as defects in proteins such as AID, which can lead to B-cell intrinsic hyper-IgE syndromes, as well as common variable immunodeficiencies. If we have a defect later that can lead to mutations that prevent class switching to IgA, and the exact genetic cause for this is still unknown. So the next group of immunodeficiencies that we'll talk about is deficiencies in cytokines. In particular, this really affects Th1 and Th17 responses. And as we'll see, there's a bifurcation between this pathway, signaling pathway, and how it goes to Th1 or Th17. And the cytokines in this, such as IL-12 or IL-23, are very important. So for the Th1 response, we see patients that are vulnerable to Mycobacterium, Salmonella, and Listeria. And these pathogens are interesting because they're all intracellular pathogens that live inside of macrophages. So one of the possible genetic defects in this response is in interferon gamma, and this leads to impaired NK cells, ILC1s, and Th1 cells. There are also defects involved in interferon signaling, so this would have a similar effect. And one example of this is STAT1. So here the STAT1 loss of function interrupts signaling of interferon gamma, alpha, and beta. So if we look at the Th17 response, these patients are susceptible to intracellular bacteria, pyogenic bacteria, as well as Candida fungal infections. And typically, these patients show mutations in IL-17, and this impairs the development of Th17 cells and ILC3s. And here actually, interestingly, we see a STAT1 gain-of-function mutation, and this can disrupt type 3 immunity. And this is because it actually leads to an excess of signaling, and that can impair Th17 development. So this is figure 13.8 in your textbook. Again, you don't need to memorize all of these diseases, but this provides a really nice summary of diseases and the type of phenotypes and mutated genes that are there. One that I would like to point out is the last one on this slide, or Apoced Syndrome. And this is actually an apopsed or error defect that leads to neutralizing antibodies. So in this case, we actually have an autoimmune problem leading to neutralizing antibodies, but these antibodies then disrupt cytokine signaling, leading to an immune deficiency. So the last group we'll talk about is just a couple miscellaneous other genetic defects that lead to interesting immunodeficiencies. So first we have X-linked lymphoproliferative syndrome, and this is typically caused by a mutation in SH2D1A or XIAP. And this leads to defective coupling of the immune receptor and tyrosine kinase, which is actually interestingly marked only by an increase in susceptibility to Epstein-Barr virus. And this is because with this genetic defect, EBV cannot establish a latent infection as it normally does. And this leads to B cells infected with the virus constantly proliferating. There are other possible defects that can also occur with this, which involves no NKT cells, as well as an impaired T-dependent antibody response. So I mentioned that this genetic defect is due to problems in the coupling of the receptors and receptor signaling. So we can see here the genetic defect prevents the tyrosine kinase from being able to signal, even if the receptor is engaged. And we can also see then impaired fast-mediated apoptosis, and this can also exacerbate the lymphoproliferation. So another immunodeficiency is Hemophagocytic Lymphohistiocytic syndrome. So this again has a really long name, but it essentially is marked by uncontrolled activation and expansion of CD8 T cells and macrophages. And these T cells, as you guys will remember from previous lectures, cause cell death and phagocytosis. So if you have uncontrolled expansion of these cells, they will enter the tissue and kill normal, healthy tissue cells, causing necrosis. And typically, these cells actually cannot kill infected target cells. So these cells actually cannot kill infected target cells, although interferon gamma release by these CD8 T cells and NK T cells is not affected. We do see increased inflammatory cytokines, and this disease is caused by a defect in delivery of cytolytic granules to target cells. So this is why we see a decrease in cell killing of target cells while also having an active expansion of these cells. And some of the genes that this defect can be in are perforin, as well as several other genes that you guys can review. So we also see in this disease that also non-immune lysosome function can also be affected. And this is again due to problems with granules within cells. And here you can see an example. So this is figure 13.9 in your textbook. So we can see T cell activation, and then perforin would normally be released. So we can see a docking with the target cell, priming to dock, and normally the cell would fuse and release the cytotoxic granules. And you can see here they've highlighted in red the specific genetic defects that will interrupt each step of this process. So there are also a few complement defects, and essentially you can have a defect in any part of the complement cascade. So this figure 1311 shows a summary of these different defects. So again, that's summarized in this table as well. And complement defects actually show increased tissue damage from immune complexes and the inability to clear extracellular bacteria. So there's actually, as I mentioned before, there are some genetic defects where there might be a problem with the gene or a protein, but there's no phenotype seen in a patient. And C2 is actually a very good example of this. So this is can be redundant in immune complex formation, and so you can actually live a completely healthy life while having no C2. And our last group of immunodeficiencies is phagocyte defects. And here, again, there's a variety of defects, but I just want you guys to think about the function of phagocytes. So the phagocytosis and killing of cells, and think about how defects in those cells could lead to immune problems. So as you might expect, this leads to an increased susceptibility of bacterial infections, and the defects can be anywhere in the development and functional pathways in these cells. There's also a small group of autoinflammatory diseases, and this is actually inflammation and fever in the absence of an infection. And one of these examples, there's a disease called familial Mediterranean fever, and this is a defect in pyrin. We also see Nlrp3 defects, and this is part of the inflammasome, which responds to cold and stress. So these two defects have really helped scientists understand fevers and stress. So pyrin is found to activate the fever response. So the Nlrp3 defect, we see episodic fevers, and these fevers may actually be induced by exposure to cold. And this makes sense with the function of Nlrp3, since it can activate the inflammasome in response to cold and stress. So a few treatments we'll talk about. This will be very brief. We're going to cover it a bit more in the lecture on biomodulators, but I just wanted to touch on it here. So some medical treatments, such as for SCID, is you can actually do a bone marrow transplant. And this replaces the defective hematopoietic cells, so these patients will develop functional B and T cells. However, the donor and the host must share some HLA alleles, and this is because the T cells must be selected on the host cells, or else the T cells will not be functional. And it's also possible to have graft versus host disease, where the bone marrow that's implanted in the patient actually begins to attack the patient. This is typically reduced if mature T cells are removed before transplantation. Another example of treatments for primary immunodeficiencies are somatic gene therapies. And this is where scientists use vectors to introduce the defective gene. So the patients will then have a copy of the gene that works properly. So previous treatments here were used in SCID, and these treatments used a retroviral vector. However, some of these patients developed leukemia because the retrovirus randomly integrated the new functional gene into the genome, and sometimes this interrupted oncogenes, and so cancer occurred. Okay, so we've talked about primary immunodeficiencies or genetic problems that lead to immunodeficiency. And now we're going to be talking about secondary or acquired immunodeficiencies. So there are several things that can cause this. First, these are actually much more common than primary immunodeficiencies. So since these are not due to genetic defects, they can occur to a wider population of individuals. So there are some environmental conditions that can cause immunodeficiency, such as malnutrition. And malnutrition, actually, we see an increase in death from measles and tuberculosis. And this can partially be due to protein starvation, which will starve the antigen-presenting cells of peptides to present. And leptin, which is a hormone responsible for appetite regulation, is also important for T-cell response. So we can also have some medical situations that lead to immunodeficiency, such as removal of the spleen. And if you guys think back to previous lectures that you've had on the different organs in the body, you'll remember that the spleen is really important for filtering the blood. So patients here are predisposed to S. pneumoniae and bloodborne pathogens in general. So we can also see immunodeficiency caused by chemotherapy or immunosuppressive treatments. And these are typically used in treatment of autoimmunity or cancer. Finally, which we'll go into a little more detail about, infection can cause immunodeficiency. And the most well-known example of this is HIV and AIDS. So what is HIV and AIDS? So AIDS stands for Acquired Immunodeficiency Syndrome. And HIV actually infects CD4 positive T cells. And this is really important for how it ends up causing immunodeficiency. So the virus, which you can see a schematic here, is a retrovirus that will integrate its genome into the host genome. And this leads to a latent infection. And AIDS actually occurs when the T cell count falls below 200 in the blood. And this is considered a very low number of T cells and will lead to immunodeficiency, as we'll talk about. So in HIV, after the virus has been integrated for a while, it's possible that when the T cells are activated in response to another infection, the virus will become reactivated because the T cell is starting to transcribe more proteins. And this requires the host transcription factors NF-kappa B or NFAT. So these factors move to the nucleus when cells are activated. So you can see here, T cell activation causes these transcription factors to move to the nucleus. Then RNA is transcribed and translated, and this will include the viral proteins. So then the virus will be able to reassemble itself and release from the cell, moving on to infect other cells in the body. And HIV, and especially AIDS, leads to opportunistic infections. And in particular, patients with AIDS are more susceptible to oral candida, tuberculosis, and shingles, as well as EBV-induced B-cell lymphoma. And there are a couple other cancers which are caused by viruses that are seen in much higher levels within patients who have AIDS. And you can see here a graph of CD4 T cells that helps explain this phenotype. So Cytomegalovirus is another viral infection that can establish a symptomatic infection in immunodeficient individuals. Most people are infected with cytomegalovirus, but we don't see symptoms unless the immune system is compromised. So we can see the compromised immune system in this chart of numbers of T cells as well as the viral load of HIV. So upon infection, we have a high number of T cells shown in the green line, And these drop, and we can see that they will seroconvert, or this is where we begin to see antibodies in the blood. So with seroconversion, some of the virus is cleared and T cells can repopulate. However, over time, T cells will be activated, the virus will be reactivated, and then these cells will be killed. So there's a gradual decline of CD4 T cells in the body. And as the CD4 T cells drop, we can see HIV RNA copies increase. And eventually, these patients will have little to no adaptive immunity. And typically patients with AIDS die from an acquired opportunistic infection rather than the virus itself. So there are some treatments that have been developed recently called Antiretrovirals that can be used to treat HIV. So these drugs will target proteins within the virus, such as the viral reverse transcriptase, the integrase, and the protease. And these are viral only proteins, and we know that because typically cells don't reverse transcribe RNA back into DNA, while the virus does. And then the integrase helps integrate that DNA into the host genome. And so we can see there are several steps along the viral infection pathway that these antiretrovirals will interrupt. And this can actually help prevent infection of other cells that have not been infected. And because the HIV virus will mutate rapidly, we see more efficacy with highly active antiretroviral therapy, which is typically a combination therapy. And this will decrease the viral load, but unfortunately, antiretrovirals cannot remove all the virus that is latently infected in T cells. Because the virus is not being translated, the immune system cannot see it, and these cells will not be targeted. So we cannot cure HIV, but with the advent of antiretroviral therapy, patients now can live a normal lifespan. So in summary, today we've talked a lot about genetic mutations, which in single genes especially will cause primary immunodeficiency. And as you saw, these diseases all have a very specific set of symptoms. And studying these diseases helps us understand the mechanisms of the immune response. And it's particularly important for understanding human immunity. So in mice, we have the ability to knock out genes and see their effects on the immune response. We obviously cannot do that in humans. So these are essentially natural protein knockouts that we see. And therefore, we can understand how removing a protein will cause a genetic immune defect. So we also talked about secondary or acquired immunodeficiencies, which are caused by non-genetic mechanisms. And as we talked about, this can be caused by medical treatments, such as immunosuppressive drugs, the environment, and infections like HIV. So thank you guys for listening. I hope this lecture helped tie together some concepts we've seen in other lectures about T and B cell development and function. And in the next few lectures, we'll be talking about autoimmunity and biomodulators. So thank you.